Hello everybody, Streptococcus pyogenes. Today we are going to discuss about this organism, and we have discussed this earlier in our class itself. As per your request on comment section, I am again presenting you the cultural characteristics, morphology and virulence factors of Streptococcus pyogenes in this presentation. So the first thing, Streptococcus pyogenes. It is named after its morphology. Streptos means twisted or coiled. So the Streptococcus pyogenes is the normal flora of human upper respiratory tract and in animals. Some of the Streptococcus pyogenes may be pathogens, causing pyogenic infections with a tendency to spread unlike the staphylococcal infections. It produces non-suppurative lesions and then acute rheumatic fever and finally glomerulonephritis, which occurs as a sequence to the infection. So then morphology, it is ovoid to spherical in shape, gram-positive cocci, which are generally arranged in chains or pairs. So chain formation is mainly due to the division of the cocci in only one plane, where the daughter cells fail to separate completely. So generally the chains are longer in liquid media than in solid media. They are non-motile and non-spore forming. They are also capsulated. Their capsules contain hyaluronic acid. So the next important thing is cultural characteristics of Streptococcus pyogenes. They are aerobic and facultative anaerobes. So in the absence of oxygen also they can grow. Their optimum temperature for growth is 37 degrees Celsius, and growth occurs only in the medium which contains fermentable sugars, or else the medium which is enriched with blood or serum. So in blood agar, the Streptococcus pyogenes colonies are small, circular and semi-transparent in nature, and it produces a wide zone of beta hemolysis. Here the growth and hemolysis can be accelerated by giving 5 to 10% carbon dioxide. The virulent strains, that is the pathogenic strains which has been isolated from the lesions, produce matte colony, whereas the avirulent strains produce glossy colonies. Mucoid colonies are formed by strains that produces large capsules. So in case of liquid media, in glucose or serum broth, their growth occurs as a granular turbidity with a powdery deposit and no pellicle is formed. So the next thing, biochemical reactions, that is their biochemical characteristics. They are negative to catalase test, bile insoluble, and they ferment sugars producing acid but without any gas. PYR test is positive, that is they can hydrolyze pyrrolidonyl beta-naphthalamide due to the presence of peptidase enzyme. So the resulting naphthalamide produces a red color upon addition of 0.01% cinnamaldehyde reagent. And the Streptococcus pyogenes doesn't ferment ribose. Then coming to the resistance, the Streptococcus pyogenes is a delicate organism. It can be easily destroyed by heat. At 54 degrees Celsius for 30 minutes, this temperature is enough for the organism to destroy. This organism is sensitive to basic resin and it has developed a very less resistance to drugs when compared to that of Staphylococcus aureus, as they are multi-drug resistant. So the organism dies in few days in culture unless it is stored at 4 degrees Celsius, preferably in Robertson cooked meat medium. And they are rapidly inactivated by commonly available antiseptics and many. The next thing is the antigenic structure of Streptococcus pyogenes, which contributes more to its pathogenicity. So the first thing is capsular hyaluronic acid. The capsular hyaluronic acid present in the Streptococcus pyogenes is non-antigenic, as the hyaluronic acid is identical to that found in human connective tissues, and hence the bacteria can disguise themselves with an immunological self-substance. It has very weak antiphagocytic activity, so it protects the streptococci against the immunological attacks by the host system. So the next important thing is the cell wall. The cell wall of Streptococcus pyogenes consists of three layers. One is the outer layer, middle layer, and inner layer. So the outer layer consists of protein and lipotecoic acid. The middle layer consists of group-specific carbohydrate, whereas the inner layer consists of peptidoglycan. The cell wall enhances non-specific resistance. like the pyrogenic and thrombolytic activity, and it is responsible for the cell wall rigidity. So this picture shows various virulence factors of Streptococcus pyogenes like the T protein, F protein, M and M-like protein, streptolysin S, and some enzymes and hyaluronic acid capsules and the peptidoglycan cell wall. So the next one is the group A specific carbohydrate. Here the serological grouping of streptococci is done on this basis. So based on the group A specific carbohydrate, the Streptococcus pyogenes is further divided into 20 Landsfield groups and it is named as a Streptococcus pyogenes A, B up to Z except I and J on the basis of the group specific carbohydrates. So all the streptococci except Streptococcus viridans have a layer of carbohydrate. So the next important antigenic factor are proteins. So the proteins are present in the outermost layer. It produces surface protein antigens like F protein, M protein, T protein and R protein. And these proteins are useful in serological typing of the F protein of Streptococcus pyogenes recognizes the host organism's fibronectin. So the fibronectin is a matrix protein that is present in the eukaryotic cells. It helps in attachment together with the lipotecoic acid and M protein. And the next one is M protein. So the M protein is the most antigenic. It acts as a virulence factor by inhibiting the phagocytosis. It is covered with a lipotecoic acid that enable the organism to attach the epithelial cell of the host. So the most distal part of M protein shows extensive variability among the strains. Hence, the persons may suffer from recurrent Streptococcus pyogenes infection with strains expressing different versions of M protein. So, on the basis of this antigenic difference in M protein, Streptococcus pyogenes can be divided into 100 more groups. So, the next thing is the T protein. It is common to many M type of proteins. It is not associated with virulence and it is not a protective antigen, but it's strongly antigenic. The next thing is R protein. It is a non-type specific and it is associated with the M proteins of types 2, 3, 28 and 48 and it is also known as M associated protein. It is not associated to virulence and not a protective antigen. It is also strongly antigenic. The next important antigenic factor is pili fimbriae, so which is most common in many of the pathogenic bacteria. So the pili is a hair-like or projections from the capsules. It consists partly of M protein and it is covered with the lipotecoic acid. It helps in the attachment of streptococci to the epithelial cells of host. So the next important antigenic factors are toxins and enzymes. Hemolysins and erythrogenic toxins are the toxins produced by Streptococcus pyogenes, whereas some other enzymes are also produced by these Streptococcus pyogenes. So the first toxin, hemolysins. The hemolysins specifically produced by Streptococcus pyogenes are said to be streptolysins. So there are two types of streptolysins and they produce complete disruption of RBCs. It contributes to tissue invasion and destruction of the host cell. So the two types of hemolysins, that is streptolysins, are streptolysin O and streptolysin S. So the next thing is erythrogenic toxin. The erythrogenic toxin is also known as pyrogenic exotoxin, DIC or a scarlatinal toxin. So it is known as erythrogenic because the intradermal injection of this toxin into the susceptible individual produced erythematous reaction, that is called DIC test, and this DIC test is used to identify a person who is susceptible to scarlet fever. The primary effect of the toxin is production of fever, hence also called as streptococcal pyrogenic exotoxin, that is SPE. So there are three types of SPE or there, A, B and C. So the immediate production of rash, for example, the rash seen in scarlet fever. So super antigens, hence massive release of cytokines occur that leads to variety of clinical signs including inflammation, shock and finally leads to organ failure. So the figure shows the rash in the scarlet fever. So the next important thing is streptokinase, which is a fibrinolysin. Here two types of streptokinase were noted, A and B. They are antigenic protein. It promotes the lysis of human blood clot by converting the plasminogen to plasmin. The fibrinolysin produced by the Streptococcus pyogenes facilitates the spread of infection by breaking down the fibrin barrier of human being around the lesions, and it is also known as a spreading factor. If it is given intravenously for the treatment of early myocardial infarctions and other thromboembolic disorders, it will be helpful. So the next important enzymes are deoxyribonucleases. The enzymes specifically produced by Streptococcus pyogenes are said to be streptodornases. They are DNases. It degrades DNA. So there are four antigenically distinct DNases are there: A, B, C and D. Among them, B is the most antigenic one. It is capable of liquefying the DNA accumulated in the thick pus derived from the nuclei of necrotic cells. So the exudate in the streptococcal infection is very thin, and it is therapeutically important in liquefying localized collection of thick exudates. The demonstration of anti-DNase B antibody in the diagnosis of Streptococcus pyogenes infections when the ASO titers is low, that is the anti-streptolysin O titers. So the next important antigenic factor is hyaluronidase. The hyaluronidase enzyme produced by the Streptococcus pyogenes it breaks down the hyaluronic acid of connective tissue of the host, and it favors the spread of infection. So the hyaluronidase here is antigenic, and it also induces specific antibodies. It degrades the capsule. So apart from these hyaluronidase, so many enzymes are there like proteinase, phosphatase, amylase, esterases, NADase, C5-peptidase, lipase, and serum obesity factor. So with this, I think you have got an idea about cultural characteristics, morphology, and virulence factors of Streptococcus pyogenes. And I will see you with another one presentation. Thank you.